Hi, my name is Chen Rong. Me, Mattia, and Taikan, we are in the group to do topic 2 to explain the digestion and absorption of carbohydrates, glycogenesis and glycogenolysis. And this is our full name and student number. Okay, now I'm going to explain the metabolism. Metabolism is the energy obtained from ingested food to make human body running. Metabolism is involved in breaking down large molecules into smaller ones and building up smaller molecules to form big ones. And there are 4 stages of biochemical energy production. There are digestion, acetyl group formation, citric acid cycle, electron transport chain, and oxidative phosphorylation. In digestion, just a lot of digestive enzyme will be involved to digest the large molecules and to form smaller molecules like glucose, amino acid, fatty acid. And in the second step, the smaller molecules produced from last step will be oxidized to degrade it into even smaller ones. In the third step, the acetyl group will be oxidized to form carbon dioxide and coenzymes. The coenzymes will finally facilitate the ATP production in the last step. And now I'm going to explain more in the digestion. Digestion is the first stage in the biochemical energy production. Digestion of carbohydrates starts in mouth. An enzyme called salivary alpha amylase will catalyze and break down the hydrolysis of alpha glucosidic bonds within starch to produce small polysaccharides and disaccharides maltose. And there is no digestion in stomach, but small intestine is the site of the most carbohydrate digestion. And the enzyme called pancreatic alpha amylase will catalyze hydrolysis of alpha glycosidic bond in polysaccharides to form disaccharides maltose. Disaccharides maltose as well as sucrose and lactose will be break down to monosaccharides. Once all this finished, glycogen synthesis can start to take place. Glycogen is the large polymer of glucose that stores glucose in muscles and liver. And glycogen is the source of glucose for glycolysis and to maintain body glucose level. Glycogenesis is the metabolic pathway of glycogen synthesis from glucose. And there are 3 steps: formation of glucose 1-phosphate, formation of UDP glucose, glucose transfer to a glycogen chain. And I'm going to explain the first step. So glucose, in the energy consuming stage, glucose is firstly break down to glucose 6-phosphate by hexone kinase. And if the glucose 6-phosphate can be used 3 ways: pentose phosphate pathways, glycolysis, and glycogenesis. If glycose 6-phosphate is used in glycogen synthesis, it will be activated on by phosphoglucomutase to form glucose 1-phosphate. And by this, the first step will be finished. Then I'll pass to Mattia to explain more on the second and third step. Thank you.
Thank you so much, Tracy, for your work. I'm very crazy to talk about the introduction of carbohydrates and metabolism in the first step of glycogenesis. We will be focusing on the second and third step of glycogenesis. Let's go straight into it. The first thing that happens is the glucose 6-phosphate is acted on by the enzyme phosphoglucomase to form a glucose 1-phosphate. Basically, this enzyme shifts the phosphate group from a carbon 6 onto a carbon 1 to form glucose 1-phosphate. It is actually important to point out that this step is actually reversible. So if this glucose 1-phosphate is going to be used for glycogen synthesis, the next step is this glucose is being acted on by the UDP glucose pyrophosphorylase enzyme, which is a very important enzyme because it takes UTP and activates glucose through the attach of a uridine moiety to the glucose to form a UDP glucose. Therefore, once we have a UDP glucose, the next step is the important role of the glycogen synthase. This enzyme actually will remove the UDP portion of the UDP glucose and add the glucose to the actually a non-reducing end of the glycogen chain, as we can see in picture. Now let's imagine that we have a very, very long glycogen chain. Where you might be asking, where are the branching points come from? Well, simply there is something called a branching enzyme. Actually, this branching enzyme removes 6 to 8 glucose molecules of the terminal ends of the long chain, and it attaches to another chain of glycogen with an alpha 1-6 linkage. So basically, the branching enzyme is producing branching points. These branching points are very, very important because the more branching points there are, the more free terminal ends that glycogen actually has. It actually speeds up the rapid release of glucose. It allows the cells to access more glucose quicker and faster. A quick final step I want to talk about is the UDP and the UTP. UTP is used to activate glucose. So when the UDP is actually removed of the UDP glucose, once the glucose is actually added to a glycogen chain, the UDP glucose has to be recycled. Be recycled back to the UTP by who? By the enzyme nucleoside phosphate kinase, and this enzyme actually requires ATP energy. Okay, so we talk about the production and synthesis, but where is it actually stored? There's two places where it's stored. The first one is the liver, and the second one is the muscle. The liver, the main role of the liver is to maintain a good glucose level. It takes up about 100 grams of the liver, and it is stored in the cytoplasm. This this is actually used during fasting, and the reservoir only lasts 24 hours. A simple way to remember is fasting for a day takes 100 grams of your liver away. The second place is the muscle. The muscle is also stored in the cytoplasm. Instead of 100 grams, it weighs about 400 grams. It's basically used for energy source, and it is utilized during exercise. A simple way to remember the difference between the liver and the muscle is in the liver you have 100, while on the muscle you reach 400. And for me, that's everything. Thank you so much for listening.
Thank you, Christian and Mattia, for talking about glycogenesis. Hello, my name is Tyken Bennett, and I'll be talking about glycogenolysis, chemical equation. Now, whereas glycogenesis refers to the synthesis of glycogen from glucose, glycogenolysis instead refers to the breakdown of glycogen into glucose. In this way, they're opposite. Glycogenolysis occurs within two steps. Number one is the phosphorylation of a glucose residue. Number two is glucose phosphate isomerization. The enzyme responsible for activating the process is called glucogen, and this occurs when there is low blood sugar, like when fasting, in hyperglycemia of the liver, and due to adrenaline in muscles. Inhibition. The hormone responsible for inhibiting this process is called insulin. Glycogenolysis can occur within the liver and within the muscle. In the liver, it has the purpose of releasing glucose into the bloodstream. This will work to benefit other cells. So what happens is the glucose 6-phosphatase, which is an enzyme, removes the phosphate from the glucose 6-phosphate, and the excess glucose can then exit via the glucose transporter. Within muscles, however, glycogenolysis provides an immediate source of glucose 6-phosphate to function for muscle contraction. This is because muscle cells actually lack glucose 6-phosphate phosphatase for breaking down the glucose, as would happen in the liver. Glycogenesis and glycogenolysis must work together for the healthy regulation of blood glucose levels.